Hello, my name's Jay Anderson. Here's a photo of me. I work at the Centre for Organics Research at Southern Cross University. I want to share with you some of the work we do with fungi and plants at Southern Cross Uni. I've titled my talk Plants and Fungi, Friends and Foes, because I want to talk about beneficial fungi and also those which harm plants. After my name here, I've put some letters. These are to indicate what I've studied. I studied a Bachelor of Horticultural Science, indicated by the BHSC, Hort Science, because I've always been interested in plants. And while I was at university studying horticultural science, I learned about the fungi which cause diseases of plants. And that's what I did my PhD on the study of diseases of plants or plant pathology. So when someone mentions a fungus or a fungi, what do you think of? Do you think of a classic agaric mushroom? Is that what comes to mind? Or maybe you think of petri dishes like these ones, work with the fungi that I work on. And how do fungi interact with plants? So if you're walking through a rainforest and you see leaf litter with strands of white extending out, and you push aside that leaf litter, or you see a broken piece of wood that's all white, what you are seeing are fungi working to break down plants and recycle the nutrients back into the ground. What about interactions with living plants? So today I will talk about two of these things that we work on at SCU at Southern Cross Uni. First one being plant pathogens, and the second one being mycorrhizal fungi. So the foes and the friends of plants. What are plant pathogens? Plant pathogens cause disease on plants. There are a number of organisms which cause diseases on plants. I work mostly on fungi, but bacteria, virus, phytoplasmas and nematodes also cause diseases on plants. And why are plant pathogens important? Well, we can have diseases of roots, like of this avocado plant, where root disease affects the establishment of the plant in the field. We can have leaf diseases, like this leaf disease on a banana, which decreases the photosynthetic area of the leaf, leading smaller bunches of bananas. We can have fungal diseases which affect fruit after harvest, which mean we cannot consume the fruit. And then we can have diseases which don't actually affect the edible portion of the fruit, but which look unsightly and consumers don't like. As well as these diseases of agricultural crops, we have diseases of natural ecosystems impacting our forests and amenity trees. So the example I have here is of the jarrah forests of Western Australia. This disease is caused by Phytophthora. And then you may see these bright yellow rust spores on this lemon myrtle leaf here. And you may have seen those bright yellow rust spores on melaleucas or tea trees or on eucalypts. And what happens is the leaves are affected and defoliate off the plants, and then we get this sparse canopy in our natural ecosystems. And I want you to remember those bright yellow spores because I'll come back to them in a little while. So how do we go about addressing these problems? Firstly, we diagnose the problem. Currently, I'm working on a problem with tea tree where big patches of plants are dying. So you can see this gap here. And this obviously impacts the yield of the farm. We've been to the farm, talked to the farmers, surveyed the site and collected samples to bring back to the lab. In the lab, we clean the roots of the plants, washing them with water, and then take small portions of the affected roots, sterilise the outside of the roots and put them on artificial media to grow out potential pathogens. So here we have some roots that we've washed off and you can see some of them are dark and rotted. And here's the artificial media that we've got fungi growing on. And now that we've got these cultures growing, we can use these to identify the fungus using microscopy and also using DNA sequencing. And this is the first step we take in helping the farmer address the issue is identifying what the cause of the problem is. As well as doing field work, we also do work in the glasshouse. In our controlled temperature glasshouse, we are working on blueberry rust. So if you remember the rust that I showed you just before on eucalypts or on lemon myrtle, this is a different rust, but it has the similar bright yellow spores. In the field, the rust fungus infects the blueberry leaves, reducing the photosynthetic ability of the blueberry plant. Unfortunately, this rust fungus can also affect the blueberries themselves. And the blueberries are okay to eat, but other states do not have this fungus and will not accept blueberries with rust on them. A complication of working with rust fungi is that they cannot be grown in petri dishes. They're non-culturable. So to study the infection on blueberry fruit, we take the yellow rust spores off some leaves from an infected plant in the field, suspend them in water, soak these little filter paper discs in the spore suspension and apply it to the surface of the fruit under controlled conditions. We are tracking the progress of the disease and taking photos so that we can share those photos with blueberry growers so they know what to look for in their crops. We're also working on organic certified control options for blueberry rust in field trials. We're working with New South Wales Department of Primary Industries to test out organic products in the field. This involved finding a collaborating farmer, whose field this is here, applying treatments over the season, and then collecting and assessing leaves for the severity of rust. So here Melinda and I are assessing leaves, and we assess thousands and thousands of leaves during the season. We work closely with statisticians to make sure our work is statistically sound, and we also consider the effects of things like temperature and rainfall on the development of diseases in the field. So if you have a look at this graph here that Steve's done for us, Steve the statistician, he's plotted the severity of the leaf disease along the axes here. He's also put the maximum and minimum temperatures from the Bureau of Meteorology and the rainfall data for us. And you can see here this rain event right at the start of May really kicked on the disease, and the disease levels increased significantly after that rainfall level. So we've identified products here in this group for further testing next season. And we never just do one season's worth of data. We want to have very good confidence in our results because farmers are relying on our information. So while plant pathogens impact negatively on plants, mycorrhizal fungi are a friend of plants and enable plants to access nutrients. Mycorrhizal fungi are a specialised group whose hyphae, the thread-like structures, which we've got here indicated in pink, these penetrate plant roots and form symbiotic relationships with plants. The plant feeds the fungus carbon from photosynthesis, while the mycorrhizal fungi feeds the plant essential nutrients scavenged from within the soil. If you can imagine, we are zooming into the roots of the plants at the interface between the roots and the fungi, we can see the special structures of the mycorrhizal fungi which enable this exchange. And we have two types of mycorrhizal fungi we are studying at Southern Cross Uni: arbuscular mycorrhizal fungi and ectomycorrhizal fungi. And the arbuscules are these little things here in the cells which enable the exchange of nutrients, and then the ectomycorrhizal form these nets you can see in the navy or the dark blue around the outside of the root. Anders Glassens is undertaking his PhD studies on mycorrhizae of sugarcane. And this is to understand more about the relationship between plants and mycorrhizal fungi. Anders' experimental methods have included field sampling to gather samples, which is then stained with special dyes to make the fungi more visible when he examines them with the microscope. Anders has produced these absolutely stunning images to help us understand more about mycorrhizal fungi of sugarcane. And you can see here, we're looking right into the cells of the roots of the sugarcane plant. Right into these cells. Luke Florence is an undergraduate student who's undertaking a project looking at mycorrhizal fungi on tea tree. There are two types of tea tree, coastal types and upland types. Luke is studying the types of mycorrhizal fungi associated with each type of tea tree. He's using similar techniques to Anders and has found both ectomycorrhizal fungi and arbuscular mycorrhizal fungi on tea tree roots. This is a very cool project to be able to do as an undergraduate student. So the last thing I want to leave you with is some of the opportunities I've had because of my plant science degree. Very early in my career, I was able to present my research at the International Avocado Congress in Spain. In 2019, I volunteered with Australian Volunteers Program and undertook surveys on smallholder banana farms in southern Lao PDR. Part of that we did survey by boat on the Mekong, a very special experience for me. And one of the things I continue to enjoy is how our research can help local farmers. I hope this has given you a taste of the exciting work we do at Southern Cross Uni on the relationships between plants and fungi. And I'd like to acknowledge Anders and Luke for enabling me to share their work.